The Autism Research Institute relies on the generosity of donors like you to make this webcast possible. If you enjoy this presentation, please consider making a donation. Thank you.
Okay, well, thank you, Sid, and it's always a pleasure to be here, and I'm always so gratified to see so many parents come to our science sessions, because I know putting up with all of our complicated diagrams and our data slides is not always comfortable, but we very much appreciate you working with us to try to better understand the molecular and cellular basis for autism. So thank you all for being here. So in keeping with our theme for the science session this year, which is the web of life, the idea that you can't tweak one strand of the web without it reverberating throughout the entire system, whether it be at the microbiome level, the molecular level, the cellular level, the organism level, or the planetary level. The idea is that we're all connected and we're all, it's a continuum that we're all involved with. So I hope you've picked that up throughout the sessions this time. And in keeping with this, I would like to tell you about our recent progress and our work in two environmentally responsive, highly relevant to autism areas of research, and that is epigenetics and oxidative stress. And I'm going to be telling you about kids and moms and immune cells and brain cells, and try to keep this web of life theme going at the molecular level. I want to start out by reviewing 5 recent publications that really have changed the way we look at gene-environment interactions in autism. And this one was reviewed by Dr. Lazare yesterday, really changed our thinking about the contribution of genes and environment. And this is a study where they used monozygotic and dizygotic twins and sophisticated statistical modeling to try to understand the contribution of genes versus environment. And what they found actually even surprised the authors, because prior to this study, the thinking was autism is primarily genetic. This study, again, largest study and sophisticated methodology, concluded, in fact, that the environment contributed a greater proportion of the risk, 58%, compared to the genes at 38%. So that was a real new finding that placed much more emphasis on the environment than it had been previously. And this study was in pediatrics, another game changer. Prior to this study, this is looking at trying to define the recurrence rate among families with autism. And they had 600 families, and this is a multi-site study, biggest study to date. And prior to this study, it was thought maybe 10 or 12% recurrence rate. And this study came out with a shocking almost 19% recurrence rate, that the chance of having a second child with autism, which really places much more emphasis on trying to understand prevention. This study was not a strong study, but provocative, and that was a retrospective study, dietary recall of mothers who had a child with autism, whether they did or did not take a prenatal supplement. And what they concluded was that the women who did take a prenatal supplement had a reduced risk of having a child with autism, especially if she had a genetic polymorphism in that pathway. This was a stronger study, much larger and much more conclusive, not in autism, but looking at women who took folic acid supplements during pregnancy and the occurrence of severe language delay 3 years later. And they found that the women who did take the supplements had a significantly reduced risk of having a child with a severe language delay. So this sort of brings in maternal diet into neurodevelopment. And then this final study was more of a review paper, But it was looking again at this idea of prenatal nutrition, epigenetics, and in this case, the risk of schizophrenia. And they proposed that the maternal prenatal nutrition can influence the risk of schizophrenia via epigenetic effects. And we're going to talk about that in terms of autism. This is a slide from that paper that shows the concept of maternal prenatal nutrition affecting folate and methionine and methyl metabolism. And of course, that is very dear and near to my heart because that is the pathways that we study in the lab and that we're going to be talking about today. And they're showing that the prenatal nutrition can affect this metabolism, which can affect the epigenetics of the fetus, neurodevelopment, and autism risk. In other words, environment mediating the outcome, the maternal environment. So in terms of gene-environment interactions, I think the field of autism, or the research into autism, has been driven primarily by this DNA to RNA to protein paradigm. And what I'd like to do today is to take you to the next step, the products of DNA, RNA, and protein, which are the metabolites that they produce. And it turns out that these metabolites actually turn around and become the building blocks for DNA, RNA, and protein. And they create the biochemical pathways that then underlie biological processes, including neurodevelopment and growth and cell function. So we're going to concentrate, and this is my area of research, is the metabolic pathways, the metabolites, and the biochemical pathways that may be altered in kids with autism, and how that might give us clues into genetics, the genetic background of that individual, and their environmental exposures. And here we have the pathways, and I'm not going to go into any detail. I just basically wanted to show you this to give you an idea of how they're interconnected. Again, part of this web concept. There are three pathways here that are clearly interdependent and interconnected. And again, these are the pathways and the metabolites that we study in our lab. So let me just walk you briefly down so you can see why we think these are such core pathways and why we think they're so critical for development. So the first pathway is called the folate cycle, and THF stands for tetrahydrofolate, and that's the form that the vitamin, folic acid that you take in your vitamin pill is used in the body. And it is metabolized to a compound called I think I'm frozen up here. Oops, I am frozen. Sorry. Anyway, exit. Well, this is not the same as it is on my computer, so I'm not sure how to do that. I don't want to make it worse. Anyway, I can keep going. So this cycle creates 5-methyl tetrahydrofolate, and CH3 is the methyl group that is then transferred to pathway number 2 through this enzyme methionine synthase. So this folate-derived methyl group, let's see if I could get that to, then is transferred through the enzyme to methyl B12, creating methyl B12, which is then the methyl group is transferred to homocysteine to make methionine. So here's this clear connection between folate-dependent 1-carbon metabolism and methionine-dependent sulfur metabolism. So once we've created methionine, it is activated to S-adenosylmethionine, which is the major methyl donor for a huge variety of essential methylation reactions. And we'll be discussing some of these, including DNA, RNA, protein, phospholipids, neurotransmitters. So this pathway, the methionine cycle, takes that methyl group from folate and transfers it to a huge number of essential methylation reactions. And then once it gives up its methyl group, it becomes SAH, which is then metabolized further to methionine, to homocysteine, which then picks up another methyl group from folate, and this cycle is ongoing 24/7 in every cell of the body, not only for ongoing protein synthesis, but also for these essential methylation reactions. And then about 50% of the homocysteine actually will leave this cycle and is metabolized down to cysteine. Cysteine is the rate-limiting amino acid for glutathione synthesis. And glutathione, if you're not familiar, is the major intracellular antioxidant and the major mechanism of detoxification. So this is a clear environmental connection here. And I'm showing it in its two forms. GSH is the active reduced form, and it donates that hydrogen to quench free radicals and to help detoxify environmental exposures. And then once it gives up its hydrogens, it becomes GSSG, which is the oxidized form, and that has to be converted back to the active form. So the ratio of the active antioxidant detoxification form, GSH, to the oxidized form is what we call the cellular antioxidant detoxification capacity. So now let me take these pathways out a little bit further and put them in a context, a functional context, so that you can see why we think these are such core pathways. So if we take the folate cycle out, it actually creates and synthesizes the bases, the purines and thymidylate, that make up the DNA. So this cycle is critical for normal DNA synthesis, for cell proliferation, for growth, for immune function. The second methionine cycle is gives us what we call our methylation capacity, and that's the ratio of the methyl donor to SAH, which is actually a product inhibitor, gives us an indication of how well that cell or that individual is able to follow through with these cellular methylation reactions. And then finally, the number 3, this is the transsulfuration pathway. This ratio of the active to inactive oxidized glutathione is what we call redox homeostasis, which is absolutely essential for normal cell functioning, for enzyme activity, for signal transduction, for the viability of the cell. So you can see that these are critical pathways. And the other point I wanted to make is that you can't, it's, again, back to the web of life theory or concept, you can't affect one part of the cycle without it feed back or feed forward affecting the others. They're all interrelated. And this is where epigenetics fits in, and we're going to talk about that a little bit more in detail so that you really, this is a not easy concept, but I want to walk you through it so that you can really understand what epigenetics is. And then we're going to talk about epigenetics in the mom and epigenetics in the child with autism. And this is where oxidative stress fits in. When that ratio gets too low, the ability to detoxify, the ability to quench free radicals is compromised, and it creates something called oxidative stress, which can lead to oxidative protein and DNA damage and cell damage. So how do we get from these epidemiologic studies to mechanism? Well, the short answer is we can't. Epidemiology does not address mechanism. It tells us that there are genes involved, that there are environmental factors involved, but the problem is that there are going to be different genes and probably different environmental factors in different kids. So we think that the genes and environment are clearly important, but independently in most cases probably not sufficient. But we would like to interject what we call the metabolic phenotype, because it is affected by both genes and environment, and I think can give us clues, again, to which genes might be relevant, which environmental factors might be relevant. And it then can take us closer, perhaps, to mechanism, an oxidative stress phenotype or an epigenetic problem with methylation, which may help us better understand the underlying issues and pathology of autism. But most importantly, we know how to fix these ratios. If they're out of balance, if they're low, they're fixable. And that gives us hope at targets for treatment and possibly even targets for prevention. And we've shown that we can, in a small study, we know that we can fix it, but what parents really want to understand is, okay, if you can fix their metabolism, does that fix their behavior? And that takes very expensive double-blind placebo-controlled studies, and we have one ongoing. I think Dr. Adams has one ongoing. We're collaborating with Bob Hendren at UCSF to answer that critical question. If we fix the metabolism, does that fix behavior? And so stay tuned, those studies are ongoing. So I'd like to talk to you about a new paradigm for autism gene-environment interaction. And that is, well, in the past, most of the studies have been done looking at the child because we know it's genetic. We look at the child's genetics and the child's environmental exposures. But we have data, and I'll be showing it to you in a few minutes, that the maternal genetics and her epigenetics and her environmental factors may be important also as risk factors for having a child with autism. And so that's a newer area, but I think it's a fertile and certainly worthwhile area to pursue, looking at prenatal environmental factors and the maternal genetics and epigenetics. So this is the best definition I like of epigenetics. It's where genes meet the environment. And epigenetics, which involves how genes control how cells control gene expression, is affected by both environment and the genes. So it really helps us understand both the genes and the environment. The genes by themselves are more or less static. They're the genetic deck of cards that your parents dealt you, whereas epigenetics and the environment are potentially modifiable. And that is really exciting because if they're modifiable, that gives us targets then for intervention and possibly even for prevention. So I'm going to talk to you quite a bit more, help you understand epigenetics, which is, again, a little counterintuitive. So the definition of epigenetics is that it's the way cells are able to turn on genes and turn off genes and maintain gene expression or maintain gene suppression. And these changes are heritable. And the tricky thing is that they don't involve a change in DNA sequence. So that makes it kind of hard to understand. We're much more familiar with genetics and mutations. And the two mechanisms that I'm going to talk to you about are DNA methylation and something called histone modifications, which are changes in the amino acid composition of histones, which are part of DNA, chromatin of the DNA. A good way to think about it is that if the genetic sequence is the hard drive, then epigenetics is the software that determines whether, how, and when that sequence will be read. And another really important aspect of epigenetics is that it's the way it is the mechanism for what we call tissue-specific gene expression, which is very important because if you think about it, every cell in the body has all the DNA to express all the cells, or to express all the genes in that DNA. But in the liver, only the genes that are important for liver function are expressed. In every organ, only in the brain, only the genes that are important for brain function are expressed. All of the other genes are permanently turned off by DNA methylation. And so this tissue-specific DNA methylation underlies how the different parts of our body know which genes to express and how to function normally. So how might it apply to autism? Well, the methyl groups that we're going to talk about, and I've showed you in our pathway, are transferred to DNA and to histones. They come from SAM, that S-adenosylmethionine in our pathway, and they are inhibited by that product, SAH. And we've shown in the past, and I've talked at these meetings, that ratio, the SAM-SAW ratio, the methylation capacity, is decreased in many children with autism. And more recently, we found it's also decreased in their mothers, which makes it very interesting. So these patterns are not random in the DNA. They're mitotically heritable throughout the cell generations, but as I said, they are environmentally responsive. They can be altered by diet, by the environment, and by oxidative stress. Another concept related to epigenetics is what we call it the ability to do what we call phenotypic plasticity. And that's the ability of the organism or the cell to respond to environmental cues. The DNA doesn't work that fast. But epigenetics, the histones, the on-off gene that I'll be telling you about, do change rapidly and allow that gene to be turned on and turned off and to activate cellular defense and whatever is required at the moment. So this slide is just to kind of help you visualize the difference. Here's DNA methylation and histone modifications. And down here at the bottom we have the chromosome, one from dad, one from mom. And as we unwind, we come to what are called nucleosomes. And this is this core of histone protein with 2 winds of DNA around it. And these little tails, amino acid tails that are sticking out, I will show you is how genes are controlled, how gene expression is turned on and turned off. If we keep going and we get to the double helix, we get up here to the single-stranded DNA, and I want to point out that it's, again, there's not random methylation. It's primarily only on cytosines is where these methyl groups are placed by specific enzymes. And here's a side-by-side comparison. We're more familiar with DNA. You have a mutation. That mutation is transgenerationally passed on. It's stable. Whereas with epigenetics, here's the DNA and a methyltransferase that's methylating that DNA. Here are the histones that get modified. And these epigenetic mechanisms are modifiable, again, as I said, by diet and by environmental exposures. And they form what we call it's a heritable form of cell memory, whether it be an organ memory, or cellular memory. There's a fascinating new approach to methylation in epigenetics, which is, could that same mechanism be involved with psychological memory? We don't know how we remember. It's certainly not a DNA-related issue. So this is really an exciting area. And again, it allows us to respond immediately and heritably to environmental exposures. And here's the same thing again, the histones, the little tails, and showing that those tails can be methylated, acetylated, phosphorylated, a whole lot. We're going to, of different modifications, we're going to focus on methylation and acetylation. And here's the cytosine in DNA, that the DNA methyltransferases transfer that methyl group. And remember, that methyl group was derived from 5-methylfolate. So this is the connection through epigenetics and folate metabolism. And then down at the bottom, I'm showing how it actually works. When a gene is turned on, that histone tail is acetylated. And what that does, it creates a electrostatic repulsion so that that DNA is in a more open configuration and the DNA is hypomethylated. And what that creates is this open configuration that allows the transcription factor to get in and express that gene. When a gene is turned off, the histones have lost that acetyl group and the DNA has become heavily methylated and the DNA compacts. And so basically, accessibility, the transcription factor can't get in, so that gene never gets transcribed and is turned off. And here it is again. This is the open configuration with the acetylated histones and the hypomethylated DNA. And here's the closed configuration where it doesn't transcribe. And here's how it's mediated is through DNA methyltransferases. There's a histone deacetylase. There's a histone methyltransferase, and then there's a histone acetyltransferase. So this is how genes then are turned on and turned off. The importance of methylation, in general, the one thing to remember is that when a gene is methylated, it's generally turned off. And when it's unmethylated by the DNA or the histones, it's turned on. And these patterns, again, are not random. They're established during embryogenesis, and again, are the basis for tissue-specific gene expression, how cells know what to do. Those genes are turned off, turned on, and all of the other genes that they could express are turned off by methylation. Also, methylation around the centromere is important for chromosome stability. When it's in that open configuration, it's more vulnerable to DNA damage. The extra X is activated by methylation, and also imprinted genes parent-specific gene expression depends on the methylation status. Some of the consequences of abnormal DNA methylation, and it's usually in the promoter region, if a gene is hypomethylated inappropriately, it can cause a gene to be expressed when it should be turned off. And conversely, if it's hypermethylated, it can be permanently turned off when it should be turned on. So when these patterns get turned awry, it can cause inappropriate gene expression that can lead to disease. Animal models has shown us that when the methylation is impaired, if you have a low methylation or hypomethylated DNA, that you can actually reactivate viral inserts. And when it's hypomethylated, it can reactivate previously methylated and suppressed viral sequences. Some of the causes that are known that can alter methylation status, clearly several polymorphisms have been associated with altered DNA methylation. Dietary deficiencies in our folate pathway and our methionine pathway, and environmental exposures, including heavy metals, valproic acid, and alcohol, to name a few. So now, with that long prologue, and I really wanted to help you get your mind around epigenetics, I want to show you the results of a paper we published a couple years ago looking at a functional polymorphism in the reduced folate carrier gene and how that was associated with DNA hypomethylation in mothers of children with autism. And this was the first indication of that gene polymorphism and hypomethylation in the moms. So what we did was to take DNA from 500 what we call case-parent trios, that's the mother, father, and child, and 500 control DNA, and we genotyped them for 5 polymorphisms, common polymorphisms in our pathway. And then we used some pretty sophisticated, I didn't, our statisticians used some pretty sophisticated statistics to determine whether that variant allele, if we saw an increase in a polymorphism, whether it was operating through the mother and or through the child. And then we looked at the maternal metabolic metabolites in her transmethylation metabolism. So just to summarize, we did not see any differences between mom, dad, or the child in 4 of the 5 polymorphisms that we looked at. But the reduced folate carrier G allele was significantly increased, interestingly, among the mothers, but not among the fathers or the case children. And we also measured their transmethylation metabolites, and they were also abnormal in the moms. And these are the data. Basically, if the child inherited 1 G allele or 2 G alleles of this RFC1 gene, there was no relative risk associated with the child's genotype. However, if the mother carried 1 or 2 G alleles, there was a highly significant increase in the risk that her child would be a child with autism. So the offspring effect was not significant, but the maternal effect was highly significant. And so what this says is, for at least this particular gene, it's the genotype of the mother, not the child, that was the determinant of risk. And this is our pathway again, and again, just to emphasize that we have a continuum here, and that a problem with the folate cycle can affect all the way over to the DNA methylation. And this is where RFC1, the gene that codes for RFC1, is the protein that carries folate into the cell. So when you have a problem, if you have a polymorphism there, that's going to decrease the availability of folate, which then will cause an increase in homocysteine and these changes in our cycle, which is exactly what we saw in the mothers of children with autism. So again, this methyl group from the folate can carry over and affect DNA methylation. So there's feed forward and feedback interaction between these pathways. So we looked then at the DNA methylation in the moms of that had the double homozygous GG allele and saw a significant decrease in what we call global cytosine methylation in the moms. the decrease in global methylation was associated with a decrease in this methylation capacity. The SAM-SAW ratio was also decreased in the case moms compared to control moms. We subsequently, just last year, published a paper in JAD looking at the same thing in the kids, methylation dysregulation in the kids, and we also looked at oxidative stress and damage in the kids as well. And this is a slide from that paper. And again, basically it's showing the same thing, that in the children, we're seeing a decrease in the SAM-SAW ratio and a decrease in the total methyl content of the DNA. Now, what that means, we really don't know yet. It may mean nothing, but it's there, and it's the first observation that DNA is hypomethylated. And what's really cool about research is that when you find something new, it leads you to yet another door. It opens one door, but then leads you to another door, and it's what keeps us going and why I'm having trouble at this point retiring. So our next study was now looking at the children, and we know that the lymphocytes are peripherally, they are globally, what we call globally hypomethylated. So we now wanted to look at what's going on with epigenetics in the postmortem brain. And this manuscript we just submitted it last week actually to molecular psychiatry, so we'll see where that goes. So just again, by way of background, remember these patterns are established very early in embryogenesis, and they're the basis for tissue-specific gene expression and how cells remember what they're supposed to do. And generally, when there is hypermethylation, that's usually associated with turning off or a decrease in gene expression. Because of the tissue specificity, it was an interesting question whether genome-wide or promoter-specific epigenetics that we see in the lymphocytes is the same as what's happening in the brain. And so this was one of our questions. I mean, is it hypomethylated in the lymphocytes? Are we going to see that in the brain as well? Are the lymphocytes a good model or substitute or surrogate for what's going on in the brain? And I'll show you the fact that they're not. So we chose to look at the cerebellum, mostly because it's the most reproducibly altered tissue in the brain, and one of the reproducible alterations are a decrease in Purkinje cell number, the neurons in the cerebellum. There's been studies showing alterations or dysregulation of GABA and glutamatergic systems. There's been studies showing activated microglia and pro-inflammatory cytokines, indication of oxidative stress. And very interesting, there have been studies looking at localized cerebellar infarcts in normal people and found that they result in deficits in attention, cognition, affective behavior, and expressive language. This is the cerebellum, and so it's interesting that it's bringing the cerebellum as possibly contributing to behavior abnormalities, not just motor, but also behavior abnormalities. This is a work of my predoctoral student, Shannon Rose, and this is so critical when you're looking at postmortem brain. You have to carefully match the case brain tissue with the control brain tissue. And we matched across sex, age, postmortem interval, race, and best we could, cause of death. And the reason this is so critical is that when you see a change in whatever you're looking at, you want to know that it's because of the autism, not because of a race difference or a sex difference or an age difference. And Shannon was heroic, actually, in doing this. She scanned hundreds of samples to get our best possible match, and it really paid off because there was no mean differences in sex, age, postmortem interval, race, or cause of death between case and control. So we picked the GRAIL2 gene. This is it's called a homeobox transcription factor, and these genes, these homeobox genes, are critical during development, and in this case in the cerebellum, to the patterning and the organization, cellular organization in the cerebellum. There's also been several family studies that have identified 2 SNPs in this gene, suggesting that it could be autism susceptibility gene. And following that, there's been mouse models. Mice that overexpress EN2 show changes similar to what we see in autism, a decrease in Purkinje cell numbers and altered dendrites. Mice that have been knocked out for that gene, that don't express the gene, also show similar changes to what we see in autism. And there's been a couple of studies looking at behavior changes when you knock out this gene. So that's how we got to looking at the EN2, the engrailed 2 gene. So when we did our global total methylation status, we got a surprise. Remember, the lymphocytes were globally hypomethylated. And we're looking now at the cerebellum, and we're seeing that in that tissue, we're seeing hypermethylation globally. The DNA is hypermethylated. It's opposite from what we found in the lymphocytes. So then we looked in the promoter region, because the promoter region methylation is really more of an on-off, whether it's tissue-specific or it's established. That's where the gene expression occurs. So we looked then to the promoter region, and lo and behold, we saw hypermethylation in 2 different assays in the promoter region of this gene. So that was kind of unusual. Actually, they were correlated, the total and the promoter region methylation. Shows that this gene and this tissue is hypermethylated. So then the next step, of course, is to look at gene expression, which was even more upsetting, because remember I told you that generally that when it's hypermethylated, the gene is turned off. Well, look what we're seeing. We're seeing an increase in gene expression and an increase in protein levels, which really kind of put me in a spin that it's, again, not what you would expect traditionally in other tissues. Hypermethylation is supposed to be associated with a decrease, and we're seeing an increase not only in the gene expression, but the protein levels. So in desperation, we went to the histones, and actually that's where we got some good news, paid off. We looked at 2 histones. This is H3 lysine 27, and trimethylation of this histone is associated with downregulation, and H3 lysine 4 is associated with upregulation. So when we looked at the one that's associated with downregulation, H3K27, it was decreased in the autism, in the EN2 gene, which is consistent with an overexpression. There's less suppression, there's less turnoff in the autism compared to the controls. And when we looked at the histone that's associated with upregulation, we saw an increase. So it looks like the histones, which are really more of an on-off signal, is overriding what we would think that, and there's been now several studies showing that, yes, in fact, that can happen, that the histone methylation can override DNA methylation in terms of gene expression. So we conclude then that we have this unusual upregulation in the presence of DNA hypermethylation in the promoter, but we think it may be the upregulation may be due to the histone changes as more so than the DNA changes. The important point about EN2 is that in animal studies, they've shown that normally EN2 is downregulated at birth. It's supposed to be downregulated. So what we're seeing in these brains of individuals with autism is that it didn't get turned off. It's still turned on. And that may give us some clues and insights into the cerebellar abnormalities that we see in children with autism. So we're very happy, finally, after we looked at the histones with what we found. But it's very complex. And the fact that we're not seeing the same thing in the lymphocytes as we are in the brain suggests that you can't look at immune cell epigenetics and assume that the same thing is going on in the brain. At least in our hands, in this gene, you can't. In fact, I doubt that EN2 is even expressed in immune cells. So that's a take-home message, is that epigenetics are tissue-specific. They're intended to be tissue-specific. And what we found is that peripheral cells are not the same thing as brain cells in terms of their epigenetics. So finally, I want to show you, this actually is not in the handout, I wasn't sure about the time. I really want to show you this data. That this was just accepted for publication in Translational Psychiatry. And we're looking now, again, at the children, but looking at the bottom half of our pathway, oxidative stress, the glutathione redox status, and looking at markers of oxidative damage and inflammation in actually the same tissue that we looked at for the epigenetics. Just to remind you what glutathione, how it works and what it looks like, it's a tripeptide. It's that cysteine, that's that rate-limiting amino acid. It's the cysteine component, and this sulfhydryl group is how glutathione acts as an antioxidant and how it can quench a multitude of different free radicals and protect us against free radical damage. It's also, as I mentioned, the major mechanism of detoxification of environmental exposures. That SH group on the cysteine is a magnet for heavy metals. And there's an enzyme called glutathione S transferase that will enzymatically create a conjugate that then protects the cell, and it's broken down and excreted, and that environmental exposure is, you're protected. And this is just a diagrammatic view of the difference when you have a robust GSH-GSSG ratio and you have kind of homeostatic alteration, and there's a toxic insult. There's a dip in that ratio, but it never reaches the toxic threshold, and then it comes back, and the homeostasis comes back. However, when you have a decrease in that ratio, like we see in the moms, we see in the kids, you have what we call a fragile homeostasis. The same toxic insult will now reach that toxic threshold and can cause damage. So as background for our study of oxidative stress in the brain, I just wanted to mention that I have presented here several times looking at plasma, lymphoblastoid cells, and fresh immune cells from children with autism, that they have a decrease in this redox or glutathione antioxidant capacity. And in our paper, we showed it was associated with markers of protein and DNA damage. So in this study, now we're looking, that was in immune cells, so now we're looking at the brain, and we wanted to know whether that decrease that we see peripherally, is that's manifested in the brain, and whether we were also interested in looking at protein and DNA oxidative damage and markers of inflammation, as well as indicators of mitochondrial dysfunction. We looked in the cerebellum, and I've told you why we chose to look in the cerebellum. And in this study, we also looked in the temporal region, a region called BA22. It's in the superior temporal gyrus, and it's important for auditory processing, and it contains Wernicke's area, which is important for speech processing. So that was a natural area of interest for autism. Okay, so here are our results looking at the glutathione redox status, and let me orient you to this slide. Here on the left side is the cerebellum, and on the right is the BA22 temporal region. The open circles are the autism samples, and the closed circles are the controls. So what we see is a decrease in glutathione in the cerebellum and a similar decrease in total glutathione in the temporal region. The GSSG is that oxidized inactive form, was significantly increased in the cerebellum in the autism samples and significantly increased in the temporal region as well. So they're both decreased. The reduced is decreased, and the inactive GSSG, the oxidized form, is increased in both tissues. So clearly the ratio, as expected, was highly significantly decreased. This is the capacity to act as an antioxidant, to protect the brain cells, to detoxify, is decreased in the samples from the cerebellum, the children with autism, or actually individuals with autism, as well as in BA22. So that's kind of scary. We then looked at DNA and protein damage. 3-nitrotyrosine is a well-established marker of protein oxidative damage, and 3-chlorotyrosine is a well-established marker of an inflammatory response. And we also looked at 8-oxodeoxyguanine, which is an oxidative addict on DNA that is produced by free radical, oxygen-free radical attack. So those were our endpoints to look to see if we were actually, whether this decrease in the antioxidant detoxification capacity was associated with damage. And so this is, again, the same format, looking at the protein nitrotyrosine oxidative damage. We're seeing an increase in the cerebellum in the kids with autism and an increase in the temporal region. The marker of inflammation is increased in the autism cerebellum and increased also in the temporal region as well. So then when we looked at the DNA oxidative damage, this is our data, an increase in DNA oxidative damage in both the cerebellum and in BA22. So that's kind of scary. That's more than just a decreased capacity. This is showing there's actual damage. Again, what it means, whether it's functionally active, we don't know. But again, it's the first indication that, that there is an issue here. Finally, we looked at mitochondrial aconitase activity. And you've probably never heard of aconitase. It's an enzyme in the tricarboxylic acid cycle in the mitochondria, and it's redox sensitive. It's sensitive to inactivation by superoxide, which is a oxygen-free radical. And it's because the enzyme contains something called an iron-sulfur cluster in its active site. And if it's hit by a superoxide, it inactivates the enzyme. So if we measure aconitase activity, it's considered to be a very good indicator of excess superoxide production by the mitochondria. And this is my little diagram of the mitochondria and how it works. Here's the TCA cycle. Here's aconitase converting citrate to isocitrate. And here's this iron-sulfur cluster. So when superoxide hits that, it bumps off one of the irons. And when it bumps off one of the irons, that enzyme doesn't work anymore. But that iron actually is liberated. It can react with hydrogen peroxide, also released in the mitochondria, to create the most damaging free radical called the hydroxyl radical. And together, those two can deplete glutathione in the mitochondria. And we've actually measured that in lymphoblastoid cells from children with autism compared to controls. You can isolate the mitochondria, which we have, and looked at the ratio inside the mitochondria, and it's decreased. So we're seeing it, again, over and over again in different cells, different tissues, different organelles. So here are results with the aconitase activity in the cerebellum, and we're seeing a decrease in aconitase activity, which is a reflection of an excessive superoxide production. We also saw a decrease in the BA22, was not quite significant, but in both areas, it looks like that this mitochondrial superoxide production is excessive. And this is a slide from our paper just showing how these may interact. When you have a low glutathione ratio, that can lead to mitochondrial dysfunction, inflammation, hydroxyl radical formation. But the point I'd like to make is it's another web example. It doesn't matter whether you start from here, it can go this way, or you start from here, it can go this way. Again, another example of a molecular web of interconnected activity. And this is where all of our, I won't show you how, but this is where all of our endpoints interact, leading to our evidence of oxidative stress and damage. So then to summarize, again, this is the largest case control study. Brain, we had an N of 15. That's huge for brain. 15 control, 15 case cerebellar samples. They're difficult to get, and they're more difficult to match. So this is, and you need numbers to be comfortable with the results. So this is the largest study that's been done looking at that. And we think that the decrease in this aconitase activity indicates excessive superoxide production and implicates the mitochondria as both the source and the target of reactive oxygen species in the brain. And the 3-chlorotyrosine is an indicator of inflammation. And we've seen one or two other papers in different ways saying that there is more inflammation in the autism brain, and this is a new way of looking at that and confirming that it appears that the brains from individuals with autism have greater inflammation than controls. So the glutathione redox status, again, was decreased in both tissues and actually correlated with that aconitase activity. And the protein and DNA oxidative damage was elevated in both, and that was associated also with a decrease in this redox capacity. So we conclude then that this extends our previous observations in the lymphocytes. So in terms of oxidative stress, oxidative damage, glutathione redox status, it looks like what we saw in the periphery, in the lymphocytes, is reflected in the brain. And it suggests that this pro-oxidant, oxidized environment is systemic. We're seeing it peripherally and we're seeing it in the brain. So the fact that we do see these parallel changes suggests then that the lymphocytes could be a surrogate because they're obviously clearly more accessible for what's going on in the brain. And then further, it results that this decrease that we see in this glutathione redox capacity may have a functional consequence. That just tells us that the capacity to detoxify, the capacity to act as an antioxidant is down. But now we're showing that there's actually maybe a consequence in terms of an increase in inflammation, impaired mitochondrial superoxide production, and an increase in protein and DNA oxidative damage. So this is the my wall poster in the office, which is every metabolic pathway in the cell. And in keeping with our theme about the web of life, we're tweaking one little part of it. But the assumption is it's affecting neurodevelopment, it's affecting membrane signal transduction, it's affecting DNA synthesis. It's way more than our little pathway. This is a slide from a proteomics paper showing that protein function, you can't affect one without affecting other. They're interconnected. And with the array analysis, we now know that gene expression is interrelated, that you don't just change one, you change many. And we know that the connectivity between cells, how well they communicate, is important for attention, cognition, memory, and learning. And as Dr. Navio has elegantly communicated to us, the cells talk to each other. They know what each other is doing and when they're in trouble. And as Dr. Mayer has shown us there, we know that the gut, the brain, and the immune system are not independent. The gut and the brain talk to each other, the immune system talks to the gut and the brain. Again, this theme of everything is connected, which leads to this wonderful quote from Chief Seattle in 1854. This we know, the earth does not belong to man, man belongs to the earth. All things are connected like the blood that unites us all. Man did not weave the web of life, he is merely a strand in it. Whatever he does to the web, he does to himself. All things are bound together, all things connect. And this is a picture of true miners with the canary and emphasizing that the environment impacts every strand of life. We are all connected with the environment. We are all canaries. And finally, I want to acknowledge the people in the lab, our study coordinators, our funding that allows us to continue. And these are real people, and they come to work every day dedicated to helping you guys understand what's going on at the molecular level with autism. So thank you very much.
The Autism Research Institute relies on the generosity of donors like you to make this webcast possible. If you enjoy this presentation, please consider making a donation. Thank you.